Before we started, I want to ask Yiran, do you want us to record this talk and upload it to our YouTube channel?
Yeah, sure.
Okay, thank you. Hello, everyone. Welcome to our talk. I'm one of today's hosts, Xiaotong from Northwestern University, and my research mainly focuses on the structure property relationships of 2D highlight perovskites. And today we are honored to have Yiran Yang from Caltech to share her research with us. And first, I want to thank everyone who has contributed to organizing today's talk, and a special thanks to Chonghe and Zhen, who has coordinated everything. Today's agenda mainly includes 4 parts. First, I will briefly introduce our society, and then Chonghe will introduce our speaker, who will share her exciting research on the wearable switch sensor. The talk will last 40 to 45 minutes. Please mute yourself during the talk. And then we will have the Q&A session. You can either submit your question in the Q&A chat, or you can raise your hand and you will be promoted to the panelist. So you can ask your question directly. Our society, the Martlet Society, is a non-profit organization that aims to build communication for young scholars. And we have the TMS talk for academia, the WBA talk for non-academia, and TMS workshop for skill set. And at the beginning of each season, we will have a special issue. And if you are interested in our society, you can check our website or follow us on Twitter or WeChat by scanning the QR code. And next, I will pass the floor to Chonghe to introduce our speaker today.
Yeah, thank you very much, Xiaotong. So it is a great honor to invite Yiran Yang from California Institute of Technology. So for those who don't know Yiran, so she is a PhD candidate at the Department of Medical Engineering and working with Professor Wang. So her research focuses on wearable electrochemical sensors for non-invasive interrogation of sweat and related analysis. So her representative work is was published in Nature Biotechnology, and so she is awarded the Baxter Young Investigator Award for her contribution, and also she is a recipient of the 430 under 30 last year. So let's give that time to Yiran for her talk. So welcome, Yiran.
Hi, thank you. Thank you, Xiaotong and Chonghe, for the introduction. It's great to be here to present my work. Here, let me share my screen. Okay. Yeah. Again, it's my honor to be here to present my previous work on wearable sweat sensor for non-invasive monitoring of circulating metabolites and nutrients. And as you might have known already, I'm a PhD candidate from the medical engineering from Caltech, and I'm working with Professor Wang on my projects. So currently, the current medicine always goes for a strategy of one treatment fits all. So basically, when you have any sort of disease or symptoms, you go to a doctor, and the doctor will prescribe a therapy that is universal. But in some cases, there are many variations between individuals that may cause the treatment ineffective or cause severe adverse effects. Future medicine, however, will prescribe more personalized therapy and more personalized diagnostics so that the treatment can be evaluated in real time, and then the more personalized treatment can be tailored to each individual to achieve the best outcome. So in order to realize this personalized medicine or personalized healthcare, lots of data sets are needed to be collected in people's daily lives, so you have a more continuous track of what's going on inside of your body. To achieve this, wearables are a very promising tool for this personalized healthcare. For example, pulse oximeter, heart monitoring watch, these two common wearables are commonly used to monitor heart rate and also blood oxygenation, which is very important in COVID days because, you know, like every day we're checking our pulses and oxygen level to make sure we're staying safe and staying healthy. However, those two are still mostly towards the physical detection of vital signs. There is limited chemical information achieved by these wearables. And the most prominent chemical-based wearable is the glucose monitoring patch, which is worn by many diabetic patients throughout their daily life. With these wearable devices, large data sets can be collected and they're continuously over time, and they can facilitate facilitate personalized healthcare. However, most of these chemical information we're getting is still from a very conventional blood draw or blood test done in a clinic. In order to get these molecular information, people usually go to clinics or go to their doctors and then perform a very invasive blood test. And the trips that you're traveling to the doctors and the clinics have determined that these results can only be achieved episodically. In future, what we are trying to achieve for the personalized healthcare or medicine is to have a device that can be worn continuously on your body, and either on your arm or somewhere else, and then non-invasive continuous monitoring of your body's molecular data can be achieved. So we are looking at a very interesting biofluid called sweat. It is a very interesting alternative to serum or blood because it also carries rich molecular information. As the sweat is secreted in the secretory coil, it travels along a dermal duct. And during this traveling process, various chemicals, including ions, metabolites, acids, hormones, protein, and peptides will partition into the sweat flow and further carried by the sweat onto the surface of your skin, which can be collected conveniently. And a very prospective view of our field is to have these sweat information collected and then which can be used for fitness tracking, whether you're fatigued or whether you're dehydrated, or have a more comprehensive diagnosis based on the molecular information in the sweat, and thus for health monitoring. And with all of these information collected by sweat, we can perform cloud computing and then perform continuous monitoring of the individual's health. And then in future, potentially predictive algorithm can be combined and then to assess these quantitative data, and thus preventative healthcare can be achieved by either giving informative dietary information or suggestions, or help you to keep a better, like a healthier lifestyle to avoid certain habits, etc. And that is like what our prospects in this field. So currently, the sweat chemical sensors are mostly electrochemical or colorimetric. So electrochemical chemical sensors for sweat monitoring is mostly ion selective, which means they have an ion selective membrane or and coated on top of a certain metal electrodes, and then perform analysis of the ion levels inside of your sweat. Or the sensor can be enzymatic, which means you have enzymes modified on top of the electrodes, and certain specific proteins or other targets can be analyzed by these enzymatic electrodes. Furthermore, there are also colorimetric sweat sensors, which are dye-based, which has some color indicators inside of the reservoir where the sweat will accumulate inside, and then certain biomarkers will change the color of the dye, and thus you can analyze the intensity of the change to know the concentration of certain analytes. So there are several challenges of the current wearable sweat sensors, and I'll elaborate them individually in the following slides. Firstly is the challenge of efficient sweat sampling. Most of the current sweat sensors have their electrodes or sensor directly in contact with your skin. It is very convenient because the sweat, once it's secreted, it's gonna get in touch with the sensor. However, there will be certain contamination either from the skin or from other areas that can interfere the sensing of the sensor. In addition, the evaporation of the sweat, because of the sensor is directly exposed to the sweat, and the sweat is directly exposed to the air, there can be evaporation issue over time, and then the sensor can have an overestimate of certain analytes that you're trying to detect. Moreover, because the sensor is directly in contact with the skin, the sweat will just keep accumulate underneath the sensor, and there will be a mixing of previously and newly secreted sweat, which can lead to very poor temporal resolution over time. Say that maybe you started with a very high analyte concentration and drops over time, and your sensor may still detect a very high concentration because of the mixing of the new old sweat, old high concentration with the new low concentration. The second challenge is the possibility or the feasibility of mass production of these sweat sensors. In order to have a wide application of these sweat sensors, it's very important to have them mass producible. Currently, many of the sweat sensors, especially the electrodes, are fabricated in clean room facilities and requires specialized personnel. However, and in addition to that, in order to achieve a very high sensitivity to various metabolites, there is usually requiring individual modification on top of each electrode, which can be prone to operational errors. Another challenge that we are having is the lack of multi-model sensing. Well, maybe this is not the challenge currently, but in the previous time when we were working on this project, there's a challenge of lacking of multi-model sensing, which provides both vital sign detection and the chemical information. In that case, which means that usually when there's like, for example, some of the chemical sensors, they require temperature calibration based on, because there can be some amperometric variation based on temperature and influence. So we need some of the information from the physical side to correct or calibrate the chemical sensor outcome. In addition to that, we also want to have a better or more comprehensive profile of a person's health, health condition. So that's why we want more multiplex sensing on top of the same platform. And the next challenge is the lack of evaluation of sweat biomarkers versus health conditions. So currently, the most approved or the most popular correlation or evaluation of the sweat biomarker with the health condition is that the sweat chloride concentration within cystic fibrosis patients. So in this case, the sweat the sweat chloride concentration is correlated to cystic fibrosis conditions, which means you can use the sweat chloride to detect cystic fibrosis and very conveniently. However, other than that, there's only sweat ethanol correlated to blood ethanol for ethanol detection or measurement, either for driving alcohol tests, etc. So there are very limited evaluation of other sweat biomarkers, and there are so many sweat biomarkers available. So there is a very urgent need to evaluate more sweat biomarkers for health evaluation. So our proposed strategy is to use laser engraving technique to solve the mass production challenge. Very simply, like you can just use a commercially available carbon dioxide laser engraver, and then the laser beam focused on top of the material can either perform etching or cutting of the material, thus enabling creating the creation of certain sensors, which we will elaborate later on. So we are using this laser engraving technique for mass production. As you can see here, for the chemical sensor, we use for the sensors, we use the carbon dioxide laser engraving to etch the surface of a polyimide sheet. So we are not cutting through the polyimide sheet, but only etching the surface. In that case, the etching process is a photothermal process and can break down the polyimide structure and make them recombine into some carbon structure, which we will elaborate later. And then these structures can be patterned to your design, whether they're in a curvy shape or straight shape. And in our case, we have, as you can see here, the middle ones are some enclosed area to pattern for chemical sensors, while we have like straight lines and also serpentine lines for other vital signs sensors. In addition to sensors, we can also use the engraver to cut through medical adhesives, which are double-sided, and then stack them together to create like a fully assembled laser cut microfluidics for sweat sampling. So this laser engraving technique really provides a feasible means for mass production for both the sensor and the microfluidics. So for the chemical sensor, we used raster scan, which means like you can see here, if you zoom in, you can see parallel lines scanning along the direction and then patterning an enclosed area. And specifically, we tune the laser setting so that we can get a very porous structure, and under optimized setting, you can actually get some graphene porous structure, which is really desired because it provides a huge surface to volume ratio per unit area, per unit electrode area. And also the graphene also provides a great electromobility for electrochemical sensing. So with our laser engraved graphene electrodes, we actually compare the electrode performance with other commercially available electrodes such as glassy carbon, gold electrode, and screen printed electrodes. And we can see that in the ferrocyanide solution, the laser engraved graphene electrode has the largest oxidation and reduction peak compared to other commercially available electrodes, which means that the has a great electrochemical performance compared to other commercially available electrodes. In addition, we use this graphene electrode, laser engraved graphene electrode, and we can detect the electroactive molecules directly in very low concentrations, which was in the micromolar ranges. So as a reference, most of the previous sweat sensors, they without the modification of nanomaterials, they can only detect only in the millimolar ranges. But our laser engraved graphene, because it's one-step fabrication, you can already detect the electroactive metabolites in the micromolar ranges, which is very impressive. And as you can see here, we detect various electroactive metabolites such as uric acid, tyrosine, ascorbic acid, which is the AA, and also the dopamine. And this can be achieved by a very simple differential pulse photometry, which we can discuss later. But anyways, using the differential pulse photometry method, we can directly see the individual electroactive metabolites at their own specific oxidation potential. Further on, we actually use these laser engraved graphene electrodes in various biofluids directly, such as in sweat and saliva, and comparing to other commercially available electrodes such as glassy carbon, gold electrode, and screen printed electrodes. Our laser engraved graphene electrode can directly detect both the uric acid at around 0.4 volts and also at the tyrosine at around 0.7 volts, which is very like a proof of how sensitive our electrode is compared to other commercially available electrodes. In addition, we also evaluate the selectivity of the electrodes. For example, as you can see here, we did some common interference. We added common interference metabolites such as glucose, urea, and dopamine, and ascorbic acid. And as you can see, the peak actually didn't change in addition to the peak amplitude. And furthermore, we conducted a very wide range of interference commonly present in sweat and in their respective concentration in sweat. And then you can see here there is limited change in the in our sensor readouts. While as adding of the uric acid, our sensor will give a corresponding increase. Furthermore, we also created the laser engraving graphene sensor for the vital sign detection, including the temperature sensor and as well as the strain sensor. So for the temperature sensor, we patterned a very serpentine line so that it is strain resistant, and we also tuned the setting so that it is very compact, and also the graphene structure can also help enable the detection of temperature, because at a higher temperature, the electron thermal velocity and the electron phonon scattering of the graphene can be increased, and thus enabling a temperature detection based on the resistance. Furthermore, for the strain sensor, it is we patterned the sensor in a very straight line and compact, which is more susceptible to strain. In addition to that, we make the graphene structure very porous, much more porous than the temperature sensor, so that once there is a bending, there's a contact resistance occurring inside of the pore structure, enabling a decrease in the resistance, which we can detect a strain response. As you can see here, on the left side is the temperature sensor, which is less porous and more compact, while as for the strain sensor, it's a very spread out and porous structure, which enable the contact resistance. And we characterize the temperature response as well as the strain response. And here is the respiration rate. As you can see, at rest, the respiration rate is relatively slower and also less in amplitude, while as after exercise, the strain amplitude is larger as well as more frequent. And we also characterize the long-term stability of the strain sensor. After 10,000 cycles of bending, it is still having a very great integrity, which is very desirable. Furthermore, for the mass production of the microfluidics, we also use the laser patterning instead of the conventional lithography cleanroom based settings to create inlets, reservoirs, channels, and outlets on top of the double-sided medical adhesives and stack them together. As you can see here, basically the laser engraved medical adhesive can be stacked and attached to the sensor, and furthermore, it can be directly attached to the skin, which is a very seamless integration with the sensor and with the human skin. And we further pasted the microfluidic patch on top of a human skin and performed Iontophoresis, which is a sweat induction mechanism commonly used in hospitals to generate sweat in sedentary individuals. So based on this sweat induction session, we actually observed the sweat rate based on a dye flowing inside of the reservoir and as well as along the channel. And based on the distance to travel as well as the time we recorded, we estimated the sweat rate, which is about 1.5 microliter per minute. And based on this sweat rate, we actually performed a simulation to evaluate how long it takes for the sweat to fill completely inside the reservoir, which is about 3 to 4 minutes, which means that our microfluidics can sample or harvest the sweat pretty fast, and it can help the sensor to detect the sweat concentration, the sweat in about 3 to 4 minutes. In addition to the fill-in time, some other factors we're considering is, firstly, is there a influence of the chemical reaction inside of the reservoir that can affect the continuous sensing accuracy? So, which means, well, whenever the sensor is scanning or performing a differential pulse photometry scan, there will be a molecular depletion inside of the reservoir. So when we're continuously scanning the differential pulse photometry and having the reaction continuously going on, will there be an influence of the sensing accuracy? So we performed experimental analysis based on different flow rates ranging from 0.25 all the way to 1.5 microliter per minute, which is a very common sweat rate we observed. And we didn't observe a significant decrease or variation in terms of the sensor outcome, which means this is our design is less susceptible to the molecular depletion in the reservoir. Furthermore, we also evaluate another factor, which is how long does it take for the reservoir to adjust to the new concentration once there's a new concentration applied at the inlets. So in this case, we also performed experimental study, like changing, like here you can see, we are changing the initial concentration from 20 micromolar uric acid, 50 micromolar tyrosine, and to the 80 micromolar uric acid and 200 micromolar tyrosine. It only takes about 3 minutes to reach the steady state of the new concentration, as well as backwards. So this really indicates that our microfluidic design enables a very fast refreshing time to update the sensor, which is highly desired. Furthermore, with the sensor patch as well as the microfluidics, we did a system integration, which means we have a microcontroller. We combine a microcontroller and the Bluetooth module on top of a flexible PCB and connect it to the sensor patch, and the entire system can be directly pasted on different parts of your body, including neck, forehead, back, as well as arm. And then once we paste it on the human body, we can usually send the commands to the microcontroller wirelessly, and then the microcontroller will be pre-programmed or instructed to collect the physical as well as chemical information automatically and send them wirelessly to the user interface. With this setup, we actually collected many subjects, including, like, here is an exercising session over time, and we can see the tyrosine as well as the uric acid variation over time, and in addition to this, we also evaluated whether the sensor outcome we detected, is it actually accurate. So we, along with this microcontroller-based collection of the data, we also collected the sweat manually during the session and tested the sweat, uric acid, and tyrosine concentration by high-performance liquid chromatography, which is a gold standard to detect or accurately quantify the concentration. And then we use this concentration tested by HPLC versus the tyrosine peak amplitude we got from the microcontroller output, raw output, and we can see it matches really well, which means that our sensor can detect the uric acid and tyrosine accurately. Furthermore, we're thinking about the application of our platform. When we were discussing with our clinical collaborator in UCLA, which is Dr. Xiaoping Li, Professor Xiaoping Li, suggested that uric acid is actually a hallmark biomarker for gout. So gout is a common inflammatory arthritis affecting 8.3 million adults. And then it is triggered by the deposition of monosodium urate, which is a crystallized form of uric acid deposited in joints. And that will cause inflammation, which is very painful. And usually patients with gout have exemplified elevated serum uric acid, which is larger than 420 micromolar of uric acid in serum. And furthermore, currently there is no cure of no cure for gout, and there is only a management based on uric acid lowering therapy and then prevention or dietary control. So more specifically, there is, in terms of dietary control, people like the gout patients are usually instructed to take less purine-rich food, because the excessive intake of purine can cause a accumulation of uric acid, which is a metabolic product of purine intake inside of your body. And usually for healthy individuals, if you happen to take one meal of purine-rich food, usually it's excreted outside of your body. However, for gout patients or like other hyperuricemia individuals, they tend to accumulate inside of the serum, and then which can cause various diseases, either and once it's reaching the hyperuricemia, it's gonna have a very negative outcome. Either accumulated long-term will lead to renal diseases and other diseases, or it can just kind of deposit in soft tissues and around joints and cause a gout flare-up. So in conclusion, UA is really a key biomarker for gout and other comorbidities, and it's really important to have a good sensor platform to monitor the uric acid level inside of your body. And we're using our patch to actually study and evaluate the sweat uric acid along with the serum uric acid upon urine consumption. So your purine-rich food contains various meat, fish, shellfish, and then once you have these purine-rich food intake, there can be an elevation of uric acid and serum. To actually to verify this phenomenon, we actually have various subjects coming on different days and asking them to take a purine-rich food after their fasting state. And then 2 hours later, we asked them to do a biking exercise session and used our patch to paste on their body and then monitored their uric acid concentration. We also collected their serum, which is by finger pricking before they bike at fasting state or 2 hours after purine intake. And as we can see here, on different days and on both subjects, we had observed an increase in sweat and serum uric acid after the purine-rich food intake, which kind of verify our hypothesis that the elevated uric acid and sweat and serum happens after the purine-rich food consumption. Furthermore, we are evaluating our patch in different populations, including healthy subjects with hyperuricemia, and patients with gout. And then at fasting state, we evaluated their sweat and serum concentration of uric acid, and we can see here clearly that both the serum and sweat uric acid concentration is the highest in patients with gout. And we pull all our data of sweat concentration UA and serum concentration UA together. We can see we did a very pilot correlation of the sweat UA and the serum UA, and we see a potentially promising trend of using sweat UA for as a alternative to the serum UA quantification. Furthermore, we asked one subject to have a purine intake after fasting state, and then over 8 hours, the sweat uric acid actually fluctuates in a similar pattern as well as the serum UA concentration, which is highly promising. In addition to that, we invited one gout patient on 2 days, one day with a UA lowering medication and one day without a UA lowering medication. And as we can see here, for this subject, on the day when he didn't take the medication, his sweat uric acid and serum uric acids are both higher on the day without the medication, which is a very, which indicates that our sweat UA biomarker can potentially serve as a non-invasive biomarker for gout management. And to summarize, so our work proposed a laser engraved, which is the mass reducible sweat sensor that can enable trace level metabolite detection, as well as providing multimodal sensing of temperature as well as vital signs. Furthermore, we use the laser engraving technique to pattern microfluidics non-invasively, pattern microfluidics in a mass reducible way, which enable efficient sweat sampling. Furthermore, we evaluated the sweat UA concentration as well in addition to gout, and potentially using sweat uric acid as a biomarker for gout management. And our future work, also our current work, is to evaluate on a larger scale of uric acid as a gout biomarker, as well as broadening our detection spectrum using the laser engraved graphene sensor for other sweat metabolites and nutrients. And we're also trying to integrate the on-demand sweat simulation, which is Iontophoresis, with our platform, because most of our previous study is based on exercise sweat, which is hard to apply for daily settings. And furthermore, we're evaluating our sensor toward metabolic syndrome monitoring. And here I would like to acknowledge support for my principal investigator, Dr. Wang, and as well as thanks to our clinical collaborators from UCLA and funding sources and our lovely research group. Thank you.
Thank you, Yiran, for the wonderful talk. We have many questions. Okay, first we have Yang Zijian. Okay, I will promote you to the panelists, so you can talk now.
Hello, can you hear me?
Yeah.
Thank you for the nice talk. This is Adil from University of Venice, Italy. So I was wondering about the selectivity of the sensors. Since you did the graphene and you with laser engraved and you make it like, okay, porous, maybe surface to volume ratios increased, but on the other side also, like it may creates different binaries, you know, that creates maybe surface defects that could decrease also, like that can hinder the electron transfer. So how you control this thing, and also like the selectivity is, I see like, okay, it was so selective for different uric acid and different markers. So how you think like this could be so selective?
Okay, so firstly, we're detecting electroactive molecules, and our strategy of detection is differential pulse photometry. So the mechanism of this detection strategy is that different chemicals can be oxidized or reduced at their specific reduction or oxidation potential. So basically, this specific oxidation or reduction potential can already provide a level of specificity of your metabolite detection. For example, uric acid will be oxidized at around 0.4 volts, while as tyrosine can be oxidized at 0.7 volts. So basically, we can just kind of have a certain level of selectivity based on the oxidation potential. And secondly, we also tested the common interference in sweat with respect to the uric acid and tyrosine, because many of the interference, they're not electroactive, meaning they cannot be oxidized or reduced. So that also provides a certain specificity. And another thing is that for other electroactive molecules, there's usually a separation of the reduction or oxidation potential. So if the metabolites is their redox potential is far away apart from our target potential, and that is okay for the selectivity. I don't know whether it answered your questions.
Yeah, thank you.
Okay, so next we have Aham. You can unmute yourself and ask your question.
Hello, thank you very much for the great talk. I just had a question about the strain sensor and the temperature sensor. Do you think that if there is any potential cross sensitivity in measuring temperature and the vital sign? And my other question is that should we replace the sensing part each time after each time that we use it, or kind of reusable, or it can be modified to be reusable? Thank you very much.
Great, thank you. So for the first question, so we actually patterned the strain sensor and the temperature differently, so that we patterned the temperature sensor in a serpentine way to reduce the strain influence. And we also actually tested the temperature under like a normal strain, normal skin curvature, and we actually didn't see a significant influence of the strain interfering, affecting the temperature sensor. But certainly under like a very dramatic bending, there will be influence on the temperature sensor, but we didn't observe that in the normal skin curvature. As for the strain sensor, it's actually not affected by the temperature interference that much, temperature variation that much. And for the second question is that whether our patch is reusable. So our patch is currently designed to be designed to be disposable. So basically, our patch can be worn on the skin for like 2 hours if you want to bite that hard. But currently, we're changing the sweat induction mechanism from exercise or thermal to iontophoresis. Hopefully, we can make this patch more durable, like for long-term use. And we're also working on some other strategy to make it longer lasting.
Thank you very much.
Thank you.
Next is Brock. You can unmute yourself and ask a question.
Hi. Excellent talk. I'm a PhD student at Virginia Commonwealth University. I'm actually working on some chemical sensors. I thought you gave a great explanation of a lot of things. I did have a question. So you mentioned you're working with iontopheresis for the extraction of the sweat, right?
Not in this project. For most of the sweat induction in this project, we're using exercise-based sweat. So we observe a very high sweat rate. Yeah.
Got it. Okay. I guess, I mean, that kind of negates my question. I know that for continuous monitoring applications, it's very difficult to use sweat. And I know that there is like a lot of research out there being done about interstitial fluid, iontopheresis, and, drug delivery for that kind of route. But specifically for sweat detection, I know that it's kind of challenging. So I guess if you're, haven't been using that, I guess that would be like your next direction, right?
Yeah, we're so for interstitial fluid, most of the concentration of their target metabolites are higher, which is very great. But also it is based, most of the ISF extraction is based on reverse iontophoresis, and which is like kind of like putting a current and then pushing the the metabolites, or for example glucose along with the sodium onto the surface, onto the skin where the cathode is in contact. So in that case, I haven't been personally working with reverse iontophoresis, but from what I know, it's really the volume, or the liquid volume you're dealing with is much smaller, and usually people are having like a sensing transducer directly underneath the hydrogel to actually detect the ISF metabolites. In that case, I don't think it's well, personally, I think maybe people can find a better way to help to update the concentration underneath the hydrogel, because ISF, once you perform one-time ISF, you have a one measurement, but after 2 hours, maybe the concentration still has some leftover on the same sensing substrate, so that might influence still. So that's why we're still looking more on sweat, because if we do one-time iontophoresis, like say we have 20 microliter of sweat pushing through the entire system in the patch or throughout the microfluidics, maybe next time we do another iontophoresis, the new sweat can push again the old sweat out, which means our sensor can sense a updated information from the sweat. So I personally think it's harder to deal with ISF, although the concentration is much higher in ISF.
Right. Awesome. Thank you so much. Appreciate it.
Okay. Yeah, thank you, Yiran. Nice to talk to you again, and thank you for sharing your great talks with me. And I have one question. I think for your ultimate goal for precision medicine or like personalized medicine, you really want a very precise measurement on each individual because people are very heterogeneous, right? But honestly speaking, when I was looking at your urine acid calculations, I think the CV of a certain time point could be as high as like 30%. I was wondering if you were willing to kind of comment about it, like where does this kind of noise coming from? Maybe it's from the device fabrications, or it is just the sweat mechanisms when you actually use chemical reactions to censor them. Because let's say for 30% CV, maybe you could not distinguish the, from people to people. And I'm just curious about.
Oh, wait, do you mean standard deviation?
Yeah.
Which are you talking?
I think the last couple of last two slides when you were measuring the UV from, I think both.
Okay.
Yeah, before, I think 1 or 2.
Okay, why can I not?
About the gout before.
Okay.
Yeah, go ahead.
This?
Yeah, somewhere here. Yeah.
Oh, here. So in the first graph, we actually have multiple patients. So it's not like a it's not like me, like one subject having such a variation. It's like 6 patients with gout and 4 subjects with hyperuricemia and 5 subjects with that are healthy. So this variation is not like from one person. It's more of an interpersonal variation. What we're trying to have for precision medicine, because as you mentioned, is very personalized. So we want to see like a fluctuation over time for one individual instead of like over 10 individuals. So I think this standard deviation is acceptable because it's like a population. And also this is a small scale. Hopefully we can get more subjects, of course, not in COVID times, to evaluate more about the variation across or within population. Yeah, but yeah, to summarize, basically for precision medicine, we want to see one person's fluctuation over time.
Yeah, thank you. And the second question is, when you are using the sweat sensing, do you afraid like the sweat, when they're generating sweat, the adhesions between the electronics and the, the skins will be kind of lost because, the Hamiltonian force will be lost.
That's a great question because we, the reason why we're again, so the adhesive between the skin and the patch is very important. And we have spent quite a long time to find a great adhesive for this application. So in this project, we used one adhesive, which is adequate. But recently, we kind of find a better adhesive for all these calculations, and it seems to improve the longevity of our sensor reading because peeling the harvesting is better. It's more improved with the better adhesive. So I think for this question is, yes, you need a better, like a very adhesive tape or medical tape to actually facilitate the longevity or the duration of this use. Yeah.
Okay, thank you so much.
In this project, we didn't observe because our monitoring is relatively short term, like over 1 to 2 hours. So it didn't affect that much. But if you want to extend its use, yes, you need a better adhesive.
Yeah, thank you.
Hi, Yiran. Hi, everyone. Can you hear me?
Yes.
Yeah, okay. Thanks for a nice talk. So I'm Pierrick from Swiss Federal Institute of Technology, and very interesting in general. I had just a general question more related to the measurements. So you said that if you keep your patch too long on the skin, then you might have contamination from older sweat compared to the current measurement that you want to do. And my question was related to the measurement. So do you change the patch each time you do a measurement, or you have established like a validation time, or how do you do that?
Okay, thanks for the question. So for the long-term study, for example, the monitoring of uric acid over like 8 hours, yes, we peel off the patch for each hour because we don't let the subject wear the patch for 8 hours. But for each hour, we let the subject bike for 15 minutes or more, depending on their sweat rate. And then we kind of just like, just put the patch on top of your skin like every hour, and then let them do a 15-minute biking session. So that's how we conducted our study. And as for the sensing measurement of the old sweat pushing out the new sweat, within the 15 minutes, we perform the chemical like detection like every 5 minutes or so, because we also want to collect the temperature data as well as the strength data. Within that time interval, there will be an update of sweat if there's any new concentration changing at the inlet, because we did the experimental verification of the refreshing time on the microfluidics, which takes about 3 to 4 minutes to sense a change or to fully update the reservoir if there's a new concentration at the inlet.
Okay, thank you very much.
Thank you.
All right, so I guess it is me. So thanks for the speech at first. My question is also very general. So I just want to ask you how efficient it is when you use your sweat sensor to collect physical and chemical information from a human body, because I remember you mentioned that you will ask the participant to do some sports such as the cycling, and before you do the measurement. However, what if the participant is in the static status? Are you still able to collect enough sweat level to do your analysis?
Great, that's a great question. Thank you. So in this project, most of our sweat, like the sweat patch monitoring, are based on exercise sweat, which is like a biking session. We chose this because it's relatively stable, not like running. You kind of like shake your body a lot. And then secondly, we are trying to push this patch onto a more sedentary route of sweat induction, which is iontophoresis, which I mentioned in the end in the outlook slide. So basically, we are currently working on integrating the sweat iontophoresis, which is a sedentary way of generating sweat by applying current to push a sweat-inducing stimulant. It's like either like a very common one is pilocarpine. So when this sweat-inducing agent is delivered into your body, it can stimulate the sweat gland to sweat. So that is our current project working on using this kind of like a induced sweat at sedentary condition to sense the sweat that is generated. Like, yeah, so that is something we're working on to make it more like less painful or less tiring to get the sweat out. Yeah.
Okay, thanks for the answer.
Thank you.
Hello.
Hi.
I think it's my turn. I have two questions. The first one is, first of all, excellent work. It's amazing work all around. First question is, what do you think about this mass producible, I guess, laser engraved graphene-based electrode, relative to, let's say, clean room manufactured gold electrode, in terms of, let's say, smallest line width? Do you think what is the smallest resolution that you have achieved with this method? And a related question: have you ever tried any non-electroactive biomarkers, as in using this as a base electrode and have other building blocks on top of it? This is my first question. Yeah.
Great. Awesome questions. So for your first question about the resolution, yes, there will be a difference in terms of resolution. For lithography, I think depending on the facility, you can go for like less than 10 micrometer or even lower resolution in the clean room. For us, the limiting, like the resolution, like one single spot of the carbon dioxide laser beam is 100 micrometer. So that is our resolution. So nothing is smaller than 100 micromolar in width. moreover, because for the chemical sensor, you basically need an enclosed area. So basically, it will probably like having something like larger than 100 micrometer or even larger. And also, like we also observed that with the larger electrode area, sensing area, working electrode area, like you can have a larger signal outcome with at the same metabolite concentration that you're detecting. So in that case, yes, there's a resolution limit based on this strategy. But if you're using other I'm not sure because I didn't try other types of laser. Maybe you can achieve even lower laser beam resolution at a higher wavelength, because currently the wavelength we're using, lower wavelength, sorry. The current wavelength we're using is 10.6 micrometer. Anyway, so that's the resolution problem. And then, but we're also actually having some collaboration with one industrial company to potentially make it more mass producible, like integrating all the steps with some sort of delicate machine that I'm not super quite sure of. Yeah, they're trying to integrate and then use the laser cutting to combine all the layers together efficiently. And then second question is about the detection targets. Yes, we're working on more non-electro active targets. For example, if you check our group's other papers, we actually did like a sensing of cortisol as well as some of the COVID antibodies based on this same laser engraved graphene platform. You can check out more on our website. And yeah, we did some modification on top of the graphene to make it more sensitive or tailored to get a higher selectivity or specificity to that target. Yeah. So yeah, if you want to detect non-electroactive, you need to make something different on top. Yeah.
Awesome. Thanks.
So next, maybe Kequan can raise a question.
Oh, thanks. Thank you. It's a really nice presentation, and it's really nice finally meeting you, even though it was just on Zoom. So, can you hear me?
Yeah, sorry, I can. I just can't see
Sweden. Yeah, it's pretty late. It's like 9 p.m. now, so.
Oh, okay.
Thank you. Light. Yeah, from your. So yeah, so I would like to ask things like I think your sensor is more like a photometric sensor or like an amperomic mode. Like you're not really using it as like a potential metric mode. So in that sense, what is the reproducibility of the electrode that you fabricate with your method or with your technique?
Cool. Great. So first, do you have another question? I can't hear clearly. Do you have a second question?
The second question is just like, yeah, like not really relating to this work, but the other works like you have been using potentiometer sensors. How do you make sure that you can make it without calibration, or how do you make like no calibration methods for your analyte sensing?
Okay, I'm not sure if I get your question completely, but yeah, first about the potentiometric detection. Actually, we are currently using it for some potentiometric detection, which we think is pretty good currently, but it's not published yet. So yeah, so it not only can work as an amperometric sensor, and second is
it's a calibration free like sensors can make it calibration free, or you need to like to
the reproducibility. Yeah. So first, I think your selection of the substrate is very important. So for this polyimide, we realized that we have to order from one single vendor to achieve a great reproducibility, because some other vendors, they have a very inconsistent surface quality in terms of polyimide. So selection of the film is very important to achieve a great reproducibility. But we also, based on this great vendor that we order from, we actually achieved a pretty good reproducibility from batch to batch over different days. So you can also check that out on our published paper, the data we have it, which is like batch to batch variation. So that kind of suggests a very great reproducibility. And And yeah, so I think under optimal condition, like having a great film consistency, you can have no calibration. Well, like calibration in terms of like start or end, but over time, like when you're having a sensing, you need a calibration from temperature because it's amperometric sensor, which can be affected by temperature. Yeah.
Thanks a lot. Thank you.
Thank you.
Yeah, then maybe Bihua can raise a question.
Hi. Hi, Yiran. Hi, nice meeting you. Thank you for the talk. This is really amazing. I was just telling the organizers, this is my first time meeting a Caltech student, so I'm super excited. Yeah, so I have a question around, the timeliness of the measurement, because I work a lot in the business and commercial aspects of technology. And recently, my work was related to non-invasive measurements and monitoring for a certain disease. But what we heard from a lot of users and experts was that, measurements taken from sweat or interstitial fluids, there's a delay in certain metabolites. And for some disease, it might be more important, like diabetes, and for some, like gout, in your example, maybe less important. So I'm wondering, taking metabolic fingerprinting from sweat, the delay in timing, is it some inherent obstacle that cannot be overcome and you have to look for disease that's suitable because of this, or is there any other technical, I don't know, technology or science you can use in order to solve this?
Yeah, that's a great question. So as for the delay between sweat and serum, we didn't do research on this delay, but we know that from Professor Jason Heikenfeld, they actually did a preliminary study on sweat ethanol and serum ethanol, and they kind of did like multi-subject. Well, and then they kind of like observed that the delay between sweat ethanol and serum ethanol is about like a 3 to 6 minute delay. However, this is only for ethanol, and different metabolite partition in different ways. So maybe some are directly partitioned like diffusion partition, and some are not diffusion partition, which can complicate a lot in terms of the delay time. And furthermore, as for whether there's a way to solve this, I don't think there is a solution at the current technology stage, but what we're looking at for this personalized healthcare based on this technology is based on sweat sensing, is that we're looking at more chronic diseases, and we want to see like if we can push it to a real actually continuous monitoring, like every minute or every 15 minutes, every 10 minutes, we can maybe give like an alert before the symptom starts. So sometimes the biomarker have a elevation or variation long before the symptoms occur, which can be used for this, which can be used, like in this case, our platform can be used. In addition, we also want to use this platform as a guidance in their daily life. For example, when we were talking to, when we were having the study with many gout patients, we realized that sometimes they don't even know that certain food is purine rich. For example, some type of sausage, or whatever, they don't have labels on them saying that, oh, this is high in purine, so whatever. Well, maybe the vendors, the other future producers should label these information, but currently there's a lot of like lack of nutritional info on these products. And maybe our platform can kind of like let them know, like once you have a little bite or like certain meal of these foods, you can actually know that, oh, it's gonna affect certain metabolites in your sweat, and then you can further like better make you can make a better food option or diet control to avoid certain symptoms or certain outcomes. Yeah, that's like, yeah, I don't know whether that answered your question or not. Yeah.
Yeah, that's great. Thank you.
Thank you.
Yeah, do we have more questions? So there are still a few times. Yeah, sure. So go ahead.
Hi, Yiran. Can
Hi.
you hear me?
Yes, I
Hi.
can hear you, but
Hi.
for some reason I can't see your face on here. But yeah.
Okay. It's a great talk, and I'm really excited to hear your presentation. This is Jen from University of Toronto, and I'm actually a PhD student working on the paper-based biosensor. And I see your papers previously, and I feel it's a great work. But I just feel curious. A very simple question is, other than the electrochemistry method, have you tried to integrating your patch with other detecting mechanism? For example, calorimetric, like calorimetric or chemiluminescence, other detection mechanism, because your current patch still need to transfer the data to the phone or to read out the signal. So is there any other way your group have tried or you have tried to detecting directly showing the signal?
Unfortunately, we didn't try anything other than electrochemical detection of the metabolites in our group using this patch. Probably because this graphene is pretty dark and kind of hard to have any changes in either in absorbance or
I see.
Color wise,
Because I see your page with laser engraved microfluidic chip, so I think it's actually a great platform. Probably can open a lot of other detection methods. Yeah.
And another thing we observed that I am not sure, but when we're the laser, for the microfluidics, we usually just stack them together without putting too much dye inside, but we did try, and it seems that it flows pretty well. So maybe it's a great possibility. And depending on which type of adhesive you try, you can either have a bendable microfluidic, or I think it's definitely, as you said, there might be more possibilities in terms of this application.
Okay, great, thank you. Another short question is about the mass production. A very, probably a little bit of stupid question is, if we fabricated a laser engraved microfluidic chip in a room instead of the clean room, because you mentioned the mass production in the clean room has to be done. Is it contamination very seriously if we fabricate your chip outside of the clean room? In a normal lab, and you use just a laser cutter.
Cool. So as for contamination, there's something you need to note. Firstly, is that this adhesive is cut through, and so definitely you will have some residue at the wall of the adhesive, based on this adhesive material, and whether when it's cutting, maybe there will be thermal process of maybe some other byproducts can be produced. So you should be aware of that. If you're gonna use this application, you need to choose whether it's acrylic adhesive or other silicone adhesive. You should test it out and see if it will influence the outcome. And secondly, in terms of particles or other surrounding environment, we didn't observe other dust or something falling on top, because the laser cutter is an enclosed area, and there will be suction due to ventilation. They will have a suction or filtration of the byproducts of ashes and something. They will be sucked out or blow out immediately away from the focus point. So that will enable a cleaner cut, or a residue free or whatever, in terms of, but still on the wall you will still see some sort of residue. In addition to the mass producibility, we're still working on with a company to potentially speed up the entire fabrication process, because currently we're still making these patches instead of a small laser cutter in our lab, but they're having a huge laser cutter with some spooling and stuff like that. Maybe they can help us achieve a better, or even a cleaner cut, or a better mass production efficiency. I think did I answer all your questions?
Yeah, thanks. I think it's really great to see your chip is from the lab fabrication to the end user like application. I think it's a great work. Thank you so much.
Thank you. Thank you.
Hi, Yiran, me again. I just thought of another question I wanted to get your quick comment on. So I was thinking about this kind of technology, you know, using your metabolites to diagnose or monitor disease. And I think about it as three, you know, there are three important components to it. So you need to have the hardware, which, you know, you're developing. It seems to be going well. And then you need to have the data, which is like also going well today with the data explosion and all these with, you know, Internet of Things and everything. And the third component, most important actually, is the biology of things. And I'm asking because I used to be a biochemist, and maybe with disease like, you know, diabetes or gout, that in your example, it's quite obvious what metabolite you can track. But there's so many other diseases, and I don't think the biologists have cracked it completely. There's very vague understanding of the correlation between them and the disease itself. Do you think that's going to be a bottleneck to the application of your technology in the future?
Yes, I think so. Yeah, we always kind of like wonder why there is so limited study in terms of the biology part. Not only like whether the sweat biomarker versus certain disease conditions, but also like how the sweat is partitioned. Yeah, we always want to see like if there's more study on the biology or like physiological side, if there's more studies coming out. But currently we're just kind of like, we're like, so, and there's also like a lot of metabolomic studies in terms of serum, and we're trying to see if there's any link between the sweat biomarker with respect to serum biomarker. If not, then this biomarker is abandoned, etc. So that's what we're trying to like kind of like trial and see in terms of our studies. But yeah, I think definitely like there should be more people working on this because yeah, because we see a lot of studies in saliva versus serum, which is very great, but we also want to see more in the sweat part. And yes, indeed, this is probably a very big bottleneck. And yeah, it's very hard for us to actually find out more like targets we're trying to get. Yeah.
Okay, thank you. I'll tell that to my biologist friends.
Yeah, that would be great. Yeah, I really appreciate that. Yeah.
I guess it's me. Yeah. So speaking of targets, you know, I was involved in a project where we make a saliva-based THC sensor. So you know, THC, one example of drugs that you know, we detect. You know, in your case, you see the drug response in terms of sweat, but you know, maybe there's drug secretion via sweat, or in other cases via saliva. And I think that's an excellent direction to look at, because especially when you have the non-electrolytic biomarkers capabilities, you know, testing testing that side more. I'm sure like this could be used as a good technology stack for a saliva-based, for example, THC sensor, because from our calculations, we currently use clean room fabricated gold sensors and gold electrodes, and they're very expensive, right? When we look at the cost breakdown, it's 50% of the cost of the entire sensor. So to be able to replace this tech stack with yours would be very good. Would save a lot of money, and that will further make the sensor cheaper, and people adopt technology more readily, and so on. So this is why I see your technology. It's more of a comment, I guess.
Yeah, great.
Yeah.
Thank you.
Yeah, do we have more questions for the speaker? Okay, seems like everybody got the answer answered. So it's really great to have Yiran today. And let's applause for our speaker. So it's a really wonderful talk.
Thank you. Thank you for inviting.